Yeah, okay. That's pretty broad, but okay.
And we can jump.
Yeah. All right. I'll give you a brief intro.
Yeah. Do you use the boxes or no?
Oh, we do use the boxes, yes. All right. Hey, everyone. It's my pleasure to introduce Carsen Stringer. Carsen's a good friend of mine. We met actually in grad school interviews many years ago now. So it's cool to be able to continue to collaborate and work together in this capacity. I'm personally a big fan of Carsen's group's work. Suite2p has become one of, if not the most dominant pre-processing tools for calcium imaging, and the amount that we've been able to learn through those tools is just hard to even quantify, as well as continuing to develop amazing tools beyond that: CellPose, FaceMap, Rastermap, maybe more. We'll learn here. So Carsen's a regular speaker here at NeuroDataReHack now, and is always a hit. So I'm really excited to welcome Carsen once again and hear what they're up to for the last year and throughout their work here at Janelia.
Thanks, Ben. Thanks so much for having me. Yeah, we're big proponents of open science and sharing data, and aligned with many of the missions that NWB has. So it's really nice to meet all of you and be here. I'll take questions at any time. So feel free to ask me about anything. Arika's going to come up partway through and switch over and talk about her work. I'm going to be talking about Fengtong Du's work and Lin Zhang's work. So some modeling that's not for modeling data specifically, not data processing. And I run a joint lab here with Marius Pakisaru at Janelia. Okay, so the first thing I'm going to do is give you a test. So you have to find Waldo in this picture. How many of you have played Where's Waldo before? Raise your hand. Ah, good number. Okay, so try to find Waldo here. You found him? Ah. You might have seen this slide before, though, so...
From a year ago.
Okay, so visual memory test. There you go. Okay, anyone else? Okay, so I chose a really hard one for this slide. So there's Waldo there, pretty small. Yeah, there are definitely easier Where's Waldos. So that was a hard quiz. So when you were doing this task, you were maybe, for those of you who've done Where's Waldo before, which was most of you, you were imagining kind of what Waldo looks like. You might have been doing feature detection, looking for his glasses or his hat. You might have been doing texture recognition, looking for the stripes on his shirt. That's usually how you do it, but like his shirt is hidden here, so it's a little mean. And while you were doing this too, thinking about it, you had a memory of what Waldo looked like from past experience, and there's kind of in your brain somewhere there's this compressed version of Waldo living there. That's your visual memory. And so we're interested in these questions. We're also interested in how visual computations are integrated with motor movements. Like you were making lots of saccades to also do this task. So these are the kinds of questions that our lab is interested in. And to answer these questions, we do large-scale 2-photon calcium imaging. These are zoomed-in recordings, actually, of 4 planes. This is at 30 hertz, so faster recording rate. And so this is to understand the features that V1 encodes, so kind of that first step, that feature detection step. And so for that, we want to show lots of images. We only have the mouse on the rig for like 2 or 3 hours, so to show lots of images, we have to show them really fast. So this is actually 2 times slower than the speed that we show the images to the mice. And we can show up to like 65,000 images in a single session to these mice. And you can watch the video of the mouse, and you can see this is what it looks like with the mouse's head fixed. There's a small cranial window here. Have they all seen calcium imaging data before? Okay, cool. All right. So yeah, so basically this is the setup. It's a great prep because we can record these mice We've tracked neurons across 30 days, for example, and you can come back to, you can record from the same mice for up to around a year, which is helpful if you want to use the same mice for different things. Okay, and we can also do larger scale recording. So this is like a little more of a standard field of view for calcium imaging, but we're lucky to have this 2-photon mesoscope that allows us to zoom out and get a really large field of view. And this is the raw data. It hasn't been pre-processed or anything. It's just been motion corrected. So you can see we get really high SNR on this microscope, and we can record around 50,000 neurons at a rate of 3 hertz. These are 2 depths in visual cortex, across visual cortex, that are being captured simultaneously. Yeah.
So I was just thinking about the task you showed before and the task you made us do. In the task you made us do, we were looking for features, right? Mm-hmm. But in the task you showed us, because you said there's not enough time, you're moving things, there's also motion involved, right? So you're swapping from one to the other. So I'm just curious, there is feature in motion and there's feature in just a plain image.
That's a really, really good question. So here we put gray screen in between. So we're just really trying to look at the onset responses of the neuron. You're right, though. Like, if we show images without any gray screen between, we get different responses depending on which images they are, because there's a difference in motion between the 2 images, and Ching has looked at that in the lab. But here, we're actually not asking the mouse to do a task. We're just trying to understand the features that V1 is encoding passively. Yeah. Is this good? Okay, so we're able to record primary visual cortex, so this is V1, and these are all the higher order visual regions in mice, which have not great names, but this is what they are. This is retrospinal cortex, which you might have heard of, and these are more like PPC, parietal cortex kind of areas. And so we've tried to make it really easy for the lab to do these recordings on a routine basis, and we've been able to do a lot of different studies with this microscope. So the tool that we developed to process this data is called Suite2p, and we recently released a new version, and the preprint is on bioRxiv that tells you actually what the algorithms all are in Suite2p. It's finally all documented. So you can take a look at how we extract these cells from the data. Basically, we find correlated pixels that are correlated across time, and each of these little different colored masks are the cells that we find. You average the pixels in that mask at every time point, and that gives you these traces down here. And then going forward, we just work with the traces. We don't go back to the raw imaging data for our analysis. Okay, one small side note that we've recently been working on 2-photon stimulation. And so Paul and Marius developed this method to do 2-photon raster stimulation, where basically you're recording from a large field of view, and you have the imaging laser and the simulation laser going through the same light path. So you may not have noticed, maybe it's more clear if I come back here, we're raster scanning in this large field of view. We're actually raster scanning like one ROI, the next one, and so on. And that's how you do 2-photon calcium imaging, is you're doing this raster scanning really fast along the whole field of view. And so we can do the same thing now with the imaging, also with the stimulation laser in the same light path as the imaging laser, and only unmask the stimulation laser over cell masks. And so these are the example cell masks where the stimulation laser will be on, and we can drive those neurons in these defined masks, which are found from Suite2p. And this is possible as well because of Carmine, which is a really nice newer opsin that has really can be activated with very little laser power. So we're able to do this even though we're only hitting the cell for a small period of time. This is what the simulation looks like. And this is Paul is stimulating around 12,000 neurons in this recording here. This is the average stimulation, and you can see that we're stimulating a lot of these targets here. And this is the trial average activity baseline to the time periods before the stimulation. We mask out the stimulation time because there's crosstalk, or like you can see the laser basically on the PMT, and then you have the response after after the stimulation. And here we're just stimulating a single frame. You can stimulate more frames. So we're stimulating the single frame across 300 milliseconds, which is our recording time, 3 hertz. So yeah, some of you might be computational people, theorists, like we're thinking of things to try to do with this, but we're hoping this will open up new kinds of questions we can answer about the brain by being able to do these perturbations at a large scale.
Ideally, shouldn't, this is a nice, really nice tool, right? So this is a good way of figuring out which neurons are connected to which neurons and find functional connections.
Tentatively, yeah. I mean, like, it could be disynaptic, like you don't know for sure when you do photostimulation like you would with connectomics or EFIS, but yeah, putative functional connectivity, yeah.
Yeah, so that's why I was trying to see if there are areas which were bright here, which were not X, to see if, there are other neurons. I couldn't spot too many, but I didn't spot Waldo too, so I don't know.
Yeah, so right now with this, we're stimulating a lot of neurons. What we've seen are like nearby, and this is in the preprint, it was just updated too, close to the neuron that's being stimulated, we see more activated neurons, but then we see suppression a bit further away from the targets, which people have reported before that there's this inhibition. And we have a lot of targets here, so we see a lot of inhibition actually. But yeah, we're still working on figuring out, analyzing non-targeted cells. Yeah.
How deep can the stimulation go into the brain tissue?
So it's in the same path as the imaging laser. So it's also raster scanning in the same way.
Yeah, but can you penetrate like, 100 microns? Yeah, how deep?
Oh, sorry. We could penetrate deeper. I mean, there will be more and more scattering the deeper you go. With that is it. 2 photon, you are able to go deeper than 1 photon. But we haven't tried deeper than like layer 2, 3, like 200 microns. It's a good question. Yeah. Okay. So now I'm going to tell you a little bit about mouse visual cortex and what we're studying here. Basically, we have the mice have a large visual field. They can see lots of things around them at the same time. They also have large receptive fields. So this is like an example receptive field for a cell, which is around 20 to 30 degrees of visual angle. And when we do these recordings, we're recording a bunch of cells that have a similar visual field. And then there's also we can compare that to the monkey visual field. So monkeys and us have much smaller receptive fields in our fovea of 1 to 2 degrees. This picture is not correct in terms of the visual field. I'll show you here. The visual field for humans is actually much bigger, but we only have really high acuity in the center part. Basically V1 we think is an important area because there are a lot of neurons in cortex dedicated to it. So it's about 10% of cortex in mice and 13% of cortex in macaques, just in V1 alone. So it's a lot of neurons that are dedicated to the visual processing, and so we wanted to know better what kind of model fits neural activity from V1. And so we build a predictive model of V1, and some of you might have seen these models before. Basically, we're going to use a convolutional layer in space. So we have our input, which is this image here, and we have a filter that runs along the image here. And at every position, it takes a dot product between the image and the filter, and that's the output here for each of these dots here. And if you pad, basically there's padding here with zeros, and if you pad with zeros like this, based on the filter size, you get an output that's the same size as the input, like you can see here. And in practice, you probably have seen convolutional layers. They have more than one filter, so you have multiple filters and multiple output channels we're going to fit for the neural activity. And for each neuron, so we have these shared convolutional filters across all of the neurons. And then for each neuron, we have what we call readout weights, and people have called them readout weights in the past, for example, from the Tolias lab. Basically, you have spatial readout weights, which is where is the neuron pooling spatially. And this spatial pooling is the same across all of the channels. So it's always pooling in the same spatial place, which kind of makes sense biologically, we think, too. It constrains the number of parameters, basically, we need to fit. And then for each of these channels, there's these what we call WC weights, which are these W1, 2, 3, which is how much you weight each of these channels. You sum them up to create the predicted neural response. And so we show many images to the mice, like I told you. We compute the predicted neural response is this blue curve here. The true response is black. And in practice, we often fit lots of neurons simultaneously. So you have these shared filters across neurons, and then each of these neurons has their own readout weights. And so we fit all of these neurons together using backpropagation to minimize the loss between these blue and these black curves. And you can see for these example neurons, the fit's pretty good. Yeah?
How do you decide the filters?
Yeah, so it's all gradient descent, basically just optimizing on the data directly. So people in the past have also used pre-trained models, but we find fitting directly to the data works best, especially when you have a lot of data, like in our case here.
So number of filters is a hyperparameter.
It's a hyperparameter, yeah, and I'll show varying it later too. Yeah, that's a good question. Yeah. Other questions? Okay, so we went through kind of this analysis, right? Of like we want to find the best model for V1. That's the simplest model. So we'll start by the number of layers, then we'll go through the number of channels too. Many of the models in the literature have 4 convolutional layers with these readout weights. So we tried varying the number of convolutional layers to see how many we really needed. We quantified the variance explained of these models, so basically how close the blue and the black curves are. So there's the explained variance, which you can just get from the blue and the black curves, but there's also this denominator, this signal variance, which basically each neuron doesn't just respond to stimuli, and you'll see in Audika's talk too, they respond to behaviors and other things. So each neuron has some amount of variance that's repeatable across trials, and so we normalize by that. Basically, that's how much variance we expect to explain for each neuron. So noisier neurons, there's less signal variance to explain, in a sense, and so that's why we normalize by this term. But basically, the punchline is we can explain about 73% of the explainable variance, like what variance is related to the stimuli in the population. And we can do this actually with just 2 layers. We don't need a deeper network for this. This outperforms previous models. Mainly the difference is we have a slightly more complicated readout, and we also have bigger filters spatially. So that's why our model only needs 2 layers. Another thing we showed was that having more images does help. So we increased the variance explained from about 0.5 to this point around 0.75 by having lots of images shown to the mice. And all of this data is publicly available, so you can download it and try it out too. Yeah.
So this would give you either one value or so. This is a time varying. The neuron firing is a time varying. So are you getting some sort of Poisson firing rate, which then you convert to neural firing? How is this? So this is a static image. So running through a convolution. Yeah, it would give you one.
It is just a static image. Yeah. So we're not thinking about time. We're literally and we're taking the time response of the neuron. We're converting that neuron by time trace matrix into neurons by stimuli, and just taking the optimal offset for the stimulus response, and just taking one bin of neural activity. So we just have the neural activity for each of the stimuli. And so these look like traces, but they're actually just a bunch of stimuli in order. I mean, they are in the order they're shown, but like we're not thinking about it temporally. We do use a Poisson loss, but we're not trying to model anything temporal.
So you bin it, get the average activity, and then you're predicting average activity over different stimulus.
Exactly. Yeah.
You present the stimulus at a fixed rate, right? Like a fixed time.
7.5 hertz, yeah.
So can you see like some kind of prediction on the onset of the next stimulus in B1?
You mean that's different based on the previous stimulus?
Yeah, so there may be some neurons that anticipate the coming of the stimulus, the next image.
Yeah, I think so. We do see some neurons that are active during gray screen as well. That could, and we see some of that. I don't know at a single session level if we would see that. We can see more of that to some extent when we show the same images and the same gray screen periods for many days. That's what Ching has done in her study. Then we'll see neurons that kind of tile the gray screen. Yeah. And you have neurons that are later in the gray screen that would be more associated with onsets. Yeah. But we're going really fast here in one session, and we haven't. Too hard. It will be hard to look at that with calcium imaging.
Thank you.
Yeah. Yeah, good question. Good questions, everyone. All right. Okay, so I showed you mouse. What's really nice is that there's publicly available monkey data from the Tullius lab from this Cadena et al. Study. So they showed over 7,000 images to these monkeys while they were fixating. They also used a really fast presentation rate, 17 hertz. They recorded with ePhys, not calcium imaging, but we can apply the same techniques to this data too. And we found basically that these models also only needed 2 layers to fit well. So you can see this fraction of explainable variance explained saturating. And we fit better than in their paper, they had a VGG model. And this is just an example where like using the pre-trained models doesn't work as well as fitting the model directly to the data, like I was saying. And so we see this comparison that monkey and mouse V1 both seem to be like basically 2 conv layers and then these readout weights kind of saying there's approximately like 3 nonlinearities to get to the responses of these V1 neurons. Okay, so now we varied the number of convolutional filters in the first and second layer now that we're saying, okay, we only need 2 conv layers to model each of the neuron responses. And so brighter here is better. What you can see is as we vary the number of conv1s, we don't see a big dependence on the fraction of explainable variance explained. You can see it's kind of flat, but there's a bigger dependence on how wide conv2 is, basically. So we kind of get optimal performance when conv1 can be pretty small, like 16, but conv2 needs to be like 320 to get good performance here. Which means like you have kind of a small set of basis filters, and that's actually kind of what they look like. We can visualize the conv1 filters very easily. It's harder to visualize conv2, although Fengtong has done some of that in the supplement for the paper. But basically, these are what the conv1 filters look like, these 16 filters. You have Gabor-like looking filters. You see filters at different spatial scales in the mouse and the monkey. And so you kind of only need these kind of like basis set of filters, and then there's this kind of expansion and dimensionality in COM2 and lots of different features being computed on top of that that are necessary to explain these V1 neurons that have very diverse responses. And so that's kind of our interpretation of it. Okay, and so the next thing we asked was, like we kind of want to keep simplifying the model. So first we asked how many neurons do we need to fit these models well? And we got a really surprising result here that because I think we have so many stimuli too, that basically the fraction of explainable variance explained doesn't change with the number of neurons you use to fit the model. It's basically flat for both the mouse and the monkey. So you don't need more than 1 neuron to fit these models, which is another suggestion too that these neurons are pretty distinct from each other, that they don't have a ton of shared features that necessarily help to fit the model, and also that we've shown a lot of stimuli so we can fit these single neurons really well without using other neurons to like reduce the noise during fitting and so on. And so what Fengtong did next, based on this, was to propose this kind of mini model architecture where, okay, we can fit 1 neuron at a time. This was fitting the 2 conv layer model we were fitting before. We can literally fit a small model to every single neuron that can maybe simplify things. Because here we had 320 filters, if you remember, for our full model. And so we can try to reduce the size of this COM2 if we're fitting each neuron separately. And we apply a sparsity penalty here to reduce the size of COM2. And when we do that, all right, so we don't see a reduction in performance when we fit these mini models, so 1 model per neuron. And we can look at how many COM2s each neuron needed. It's around 32 for the mouse and around 8 for the monkey. And so the mouse neurons seem to be combining more features than the monkey neurons. But in both cases, we have fewer filters now that can explain each neuron. And you can go in and you can actually look at the weights for each of these neurons and dig into it a little more. It could be interesting if you're doing like small scale recordings. These kinds of models might be helpful to you. For example, if you don't have tons of neurons in your recording too, that you can simplify things and dig in further into architecture potentially. Yeah.
Why would you expect there to be a difference by using more neurons here? Because the goal of the ConvNet was anyway to predict the activity of a single neuron. So the weights are just assigned based on predicting that 1 neuron at any instant, like the associated weights. Unless there's crosstalk between them.
Yeah, sorry. So when we're fitting this, I'll show the full model here. When we're fitting this model, these conv weights are shared across neurons, actually. So in theory, and what people have shown in the past, although it's been a small dependence in the past, is that it can help to have more neurons to fit these shared conv weights, and then the readout weights are separate. So people have thought, okay, it would be helpful to have more neurons, but we don't think it actually is. If you have lots of stimuli, you can fit single neurons very well. You don't need multiple neurons to fit these shared conv layers. Yeah. Are there any other questions? Okay, I'm going to skip over this in the interest of time, but you can also train you can train these models and look at dependencies of comp1 and comp2 with respect to tasks. And so the next thing we did was basically look, so now I've told you a lot about kind of what we think the architecture of these V1 neurons is, kind of a simplified model of them. Now we want to ask, can the mice perform texture discrimination? And also we're thinking about how they might learn visual memories of places they've been, like these textures. So I'll tell you really briefly about that, and then I'll have Audica take over. Basically, Lin, in the lab, he trained mice to discriminate the circle and leaf corridor. And in each of these corridors, there's a sound cue that's played at a random position, denoted by this purple bar here. In the circle corridor, this sound cue is not followed by reward. In the leaf corridor, it is followed by a reward. So these are complex visual stimuli. They've been frequency matched. Basically, their Fourier spectra are matched. So this is not necessarily a trivial task for the mouse to do. But here's a mouse that learned the task. It runs straight through the circle corridor. It doesn't stop to lick. Now it's in the leaf corridor. This is a mouse that's well trained, so it's going to stop at some point. It collects the reward. Now it keeps going. Okay, now here's a new leaf corridor, another leaf corridor. It's the same visual cue, but you can see the reward happen at a different time. You saw the mouse stopped earlier. So that's related to the randomness of this. And then again, it's going through the circle corridor. It will hear a sound cue, but it knows it's not rewarding, and it just runs straight through. So Lin trains these mice for 2 weeks. Before learning, the amount of licking before the sound cue is pretty similar between circle and leaf, and then after learning, there's a significant increase in the number of licks in the leaf corridors and a decrease in licks in the circle corridors. So mice can learn this task. Okay, and while we're doing these recordings, we're lucky we're able to do these large-scale recordings, and we're going to look at all of these different visual areas and how they relate to different variables in the task. So the first thing we did was just look for stimulus-selective neurons. So we were trying to kind of do the simplest analyses possible to try to see what the biggest effects on learning are on the neural activity. So this is kind of like the simplest thing we could do, compute a D' selectivity index for each neuron, which is you take the mean activity of the neuron on the leaf corridors, the mean on the circle corridors, subtract them, and divide them by the average of the standard deviation of their activity across trials. So basically, if they have high responses in the leaf corridors, for example, and that response is high on every single trial, that neuron's going to have a high D', and it's low on the circle trials. And so here's examples of those leaf selective neurons that have high D'. So we compute the D' on training trials, and here's their activity on held-out test trials, to circle trials and leaf trials. So this example neuron you can see is responding high in the leaf corridor at the end. There's different neurons that respond at different time periods throughout the leaf corridor. And we see a similar thing for neurons with a large negative D'. So these are circle selective neurons. We see lots of neurons tiling the circle corridor as well. And so we wanted to know, where are these stimulus selective neurons both before and after learning? And so I'll leave the brain map here as I show this. Basically, in primary, what we see is in primary visual cortex, there's a lot of neurons that are selective before learning, and that makes sense, right? Visual stimuli drive V1 very well. But then after learning, what we see is a really large increase in the number of selective neurons in these higher order medial visual areas. So like retrosplenial cortex, AM/PM, areas that are more connected to hippocampus. And so Lin also did this task. So this is in mice that are trained to do the task. Lin also had a cohort of mice that just ran through these corridors for 2 weeks but never received any visual stimuli. So this is basically mice that have had lots of visual experiences with these corridors, but there's no like valence associated with either corridor. And we actually saw similar changes in these mice as well, where we saw a large increase in the number of selective neurons in these higher order medial visual areas. So what this suggests is that the mice don't need to be doing a task to do visual learning. They'll learn about their surroundings just by going through these corridors. And that makes sense to you too. I'm sure for those of you who haven't been to Janelia before, you were able to walk around and figure out what things look like. You learned the landmarks throughout Janelia, and you didn't need to like take a sip of coffee or something or get a reward every step you took around Janelia. Okay, so this is kind of a summary of these plots on the right, just dividing things up into V1, medial, lateral, and anterior. We see that there's not a big change in activity in V1 before and after learning, but there's a big increase in the number of selective neurons in these medial higher visual areas, which we think is associated with unsupervised learning. And then what we did see, interestingly, were some changes related to reward in the anterior area. So there's an increase in the number of selective neurons just in the task mice and not in the unsupervised mice. So I'll show you a little bit about the anterior neurons. I won't go into more details about these neurons in the medial visual area, but we think they're doing some kind of form of contrastive learning, potentially. And we basically, Lynn showed many more stimuli, and we looked at how the representations changed in both the supervised and the unsupervised mice. I mean, you can dig into the paper for more details. I'll just show you the reward anticipation neurons. So these are the neurons that we only saw in the task mice and not in the unsupervised mice. So basically, here's how we visualize neurons and actually how we found these neurons that were related to reward. So we have a plot of neurons by time, and we use our algorithm Rastermap to put neurons that are correlated to each other and next to each other in this plot. So we find this a useful tool for visualizing the activity. Have any of you used Rastermap before? Okay, well, you have one person. All right, well, you have one more day left to try it if you want to. Basically, the code is set up kind of the same way as like a scikit-learn function. So it's easy. You do like model.fit, and you can get an embedding and visualize the neural activity if you'd like to try it. Basically, what we like about this approach is that for things that you can't easily trial average, like for example, these green lines are when the rewards happen. They're happening at different times on every trial. So we can't just average over trials to see effects. We also there's different times when the mouse licks, runs, things like that. Each of these colors is when the mouse is in each of these corridors. Rastermap finds these sequences of neurons that are activated in the corridors, in the leaf corridor, the circle corridor. You can see that these sequences stop when the reward happens. That's because the mouse stops running. So the mouse is running, you have visual flow, and then when the mouse stops, the visual flow stops, and these are visually driven neurons, they stop responding, and then they start up again once the mouse starts running. So even these neurons that we found that came up in unsupervised learning, they seem to have require these visual inputs to be well driven. Okay, so I've rambled on enough about Rastermap. Basically what we found while looking at these raster maps was this population of neurons that seemed to ramp up and then turn off right after the mouse got the reward. Just in the leaf corridor, these neurons are not active in the circle corridors. So what we think about these neurons is that there may be reward anticipation neurons, because each corridor only has one reward. So once the mouse gets the reward, there's no more reward to anticipate, and these neurons kind of turn off. And so you can see that here. These neurons were mainly in anterior areas, and we can look at their activity across trials. So this is averaging all of these neurons here and now sorting the trials by the time of the sound cue. You can see they're not very active in the circle corridors, but they seem to ramp up to the time of the sound cue in the leaf corridor, and then the mouse gets the reward right after the sound cue here. So we have different things, so visual cortex has many different signals. It has signals that are related to, I'll go to the summary slide, related to visual feature learning. We think this unsupervised learning is an example of that. And then there's also these supervised signals that are related to reward that we found. And in both of these papers, all of the data and code is shared, so you can analyze this data too and find new things that we didn't look for. Thanks everybody. Are there any other questions before we switch over? Yeah.
Thank you. Just kind of a practical one on data analysis with Suite2p. For some larger fields of view, I've done like 1024, 1024 or higher resolution images, and I found those are kind of hard to run through Suite2p. For your larger scans, do you guys break it up into smaller, like 512 by 512 sections, or do you find it does well with certain settings on larger fields of view?
Are you zoomed in as well, or no, just you're zoomed out?
Yeah.
Yeah, so we automatically are, like those stripes you saw, our ROIs are those different stripes. So we process each of those stripes individually.
I see.
So we have things that are like 400 by 1,000, for example, in the biggest case. And that seems to work okay. But if you're having issues, yeah, you might want to split it even more.
Gotcha. And then a second question is just in terms of your like newest release, because you have the sparsity model and the other one. And I was just curious if you have like kind of a simplistic explanation of the difference between those two algorithms.
Yeah, so the sparsity algorithm is looking for sparse sources in the data, and we find that works the best in most cases. It doesn't have any prior on what the shapes of the cells should look like or anything like that. Well, it's looking for correlated pixels, basically, that in these squares that we sum. And it's just it's just finding things that look like that. There's no like kind of smoothing done on the data or anything before the processing. There's some high pass filtering, but that's it. Whereas in the sorcery model, there's smoothing. There's this assumption of more circular ROIs, so that can be more useful for lower SNR data. But in 90% of the cases, I think that sparsity usually works the best. Sparsity works the best. What you can do if you're having issues is tune a little bit the high pass filtering in space. So that's like how much we high pass filter to get rid of the neuropill, the background signals. And that, if it's too small, you'll miss cells if the cells are big, or if it's too big, maybe you're not subtracting enough of the neuropill and it can be harder to find. For low SNR, also, like, I would recommend trying something like CellPose2, which is just anatomical. But yeah, that's kind of the very, very broad summary. Are there any other questions people have? Yeah, you did not get a Suite2p talk, sorry. Okay, thanks everybody.